In human chromosomes, DNA is coded by proteins to form an exceedingly long beaded string. This string is folded into numerous loops, which are believed to help cells control gene expression and facilitate DNA repair, among other functions. A new study from MIT suggests that these loops are very dynamic and shorter-lived than previously thought. In the new study, the researchers were able to monitor the movement of one stretch of the genome in a living cell for about 2 hours. They saw that this stretch was fully looped for only 3 to 6% of the time, with the loop lasting for only about 10 to 30 minutes. The findings suggest that scientists' current understanding of how loops influence gene expression may need to be revised, the researchers say. Many models in the field have been these pictures of static loops regulating these processes. What our new paper shows is that this picture is not really correct, says Anders Sejer Hansen, the Underwood Prescott Career Development Assistant Professor of Biological Engineering at MIT. We suggest that the functional state of these domains is much more dynamic. Hansen is one of the senior authors of the new study, along with Leonid Mirny, a professor in MIT's Institute for Medical Engineering and Science and the Department of Physics, and Christoph Zechner, a group leader at the Out of the Loop using computer simulations and experimental data. Scientists including Mirny's group at MIT have shown that loops in the genome are formed by a process called extrusion, in which a molecular motor promotes the growth of progressively larger loops. The motor stops each time it encounters a stop sign on DNA. The motor that extrudes such loops is a protein complex called cohesin, while the DNA-bound protein CTCF serves as the stop sign. In their new study, the researchers developed techniques that allowed them to fluorescently label CTCF DNA sites so they could image the DNA loops over several hours. They also created a new computational method that can infer the looping events from the imaging data. This method was crucial for us to distinguish signal from noise in our experimental data and quantify looping, Zechner says. We believe that such approaches will become increasingly important for biology as we continue to push the limits of detection with experiments. The researchers used their method to image a stretch of the genome in mouse embryonic stem cells. If we put our data in the context of one cell division cycle, which lasts about 12 hours, the fully formed loop only actually exists for about 20 to 45 minutes, or about 3 to 6% of the time, Gross-Holz says. If the loop is only present for such a tiny period of the cell cycle and very short-lived, we shouldn't think of this fully looped state as being the primary regulator of gene expression, Hansen says. We think we need new models for how the 3D structure of the genome regulates gene expression, DNA repair, and other functional downstream processes. While fully formed loops were rare, the researchers found that partially extruded loops were present about 92% of the time. These smaller loops have been difficult to observe with the previous methods of detecting loops in the genome. In this study, by integrating our experimental data with polymer simulations, we have now been able to quantify the relative extents of the unlooped, partially extruded, and fully looped states, Brando says. Since these interactions are very short, but very frequent, the previous methodologies were not able to fully capture their dynamics, Gabriele adds. With our new technique, we can start to resolve transitions between fully looped and unlooped states. The researchers hypothesize that these partial loops may play more important roles in gene regulation than fully formed loops. Strands of DNA run along each other as loops begin to form and then fall apart, and these interactions may help regulatory elements such as enhancers and gene promoters find each other. More than 90% of the time, there are some transient loops, and presumably what's important is having those loops that are being perpetually extruded, Mirny says. The process of extrusion itself may be more important than the fully looped state that only occurs for a short period of time. More loops to study since most of the other loops in the genome are weaker than the one the researchers studied in this paper. They suspect that many other loops will ALS. They now plan to use their new technique study some of those other loops, in a variety of cell types. There are about 10,000 of these loops, and we've looked at one, Hansen says. Using the technology platform we've set up, which combines new experimental and computational methods, we can begin to approach other loops in the genome. The researchers also plan to investigate the role of specific loops in disease. Many diseases, including a neurodevelopmental disorder called FOXG1 syndrome, could be linked to faulty loop dynamics. The researchers are now studying how both the normal and mutated form of the FOXG1 gene, as well as the cancer-causing gene MYC, are affected by genome loop formation. The research was funded by the National Institutes of Health, the National Science Foundation, the Mathers Foundation, a Pew Stewart Cancer Research Scholar Grant, the Chaires d'Excellence Internationale Blaise Pascal, an American-Italian Cancer Foundation Research Scholarship, and the Max Planck in Dynamics of CTCF and cohesin-mediated chromatin looping revealed by live cell imaging. Science, 2022. doi. 10.1126, Science. ABN 6583 Massachusetts Institute of Technology. 2022, April 14. Structures considered key to gene expression are surprisingly fleeting. Science Daily. Retrieved April 14, 2022 from ift.tt/FLCDNC Massachusetts Institute of Technology. Thanks for watching. Please subscribe my channel for more videos.